So let's go ahead and take a look at how we go about regulating when we eat, or more importantly, when we don't eat. And so what we're looking at is we're looking at a antagonistic reflex loop here, which I like to call the pull to push away responses, in which we basically are going to look at why do I feel hungry? Why do I feel full? Why is it that I start salivating as soon as I start thinking about food? What allows the make you full stuff to actually make you full? The pull to response is the I need food. The push away response is I'm full. And so you think about this as I'm pulling the plate towards me or I'm pushing the plate away from me in terms of the overall responses that we're looking at. So part of this response goes back to the regulation that we looked at in terms of motility, where we're going to have the various types of sensory input coming in from the alimentary canal, combined with the hepatic portal responses sensing 6 carbon and 3 carbon sugars. That's going to indicate relative metabolic status. We're going to have hypothalamic signals that are going to come into play that are going to be regulating endocrine responses. We have different hormones that are going to come into play that are going to be stimulating cortical functions so as to induce the want to eat or repress the need to eat. The whole entire system is higher cortical in nature. It's a cognitive response, which means that you're able to higher cortically change and modulate so as to slow down either one of the two responses that you're looking at. By slowing down either one of the two responses you're looking at, you can either increase the materials that you're consuming or reduce the duration by which you consume. So what you're able to do if you happen to be, say, fasting right now, is you're able to use higher cortical functions so as to offset the signals that are coming from the submucosal plexus and the endocrine signals saying, hey, dummy, you need energy. You need fuel sources. Because that's what the push to pull away reflexes are going to be dictating. When we look at this whole entire response, it's based off of a cluster of hormones that come into play. Here's just a small amount of the hormones that are going to be involved with the two distinctive reflexive loops. We have things like gastrin and GRP. We have things like CCK and ghrelin. We have leptin, we have insulin, we have adiponectin, we have glucagon-like protein 1, we have secretin, we have oxyntomodulin, we have endorphins, we have POMC, we have neuropeptide Y, we have PYY. We have serotonin and dopamine and endocannabinoids. All of these different signals are going to be somehow impacting the higher cortical regions that have reflexive loops within the hypothalamus, in particular within 3 nuclei within the hypothalamus, that are going to be stimulating the mammillary bodies so as to drive the two loops. The long loop is called the long loop because it takes a long time to initiate. The short loop is called the short loop because it doesn't take that long to initiate. Both of them are going to be somewhat tied with anatomical motility within the alimentary canal. However, the overall responses within the two loops is based off of my cognitive understanding of my fuel source availability relative to my energetic demand. If I have a large amount of motility going on within the alimentary canal, my relative energetic demand is low. Because my relative energetic demand is low, I tend to not need as much fuel. And since I do not need as much fuel, I'm able to inhibit the long loop and excite the short loop. However, when I have very low motility and a large amount of energy expenditure going on, I start to have a change of metabolite and hormonal signaling that is going to excite the long loop and inhibit the short loop. The short loop stops the consumption of materials off of an inhibitory feedback loop so as to ensure that we have proper time to allow for full digestion and absorption of materials. The long loop tends to be more of a stress response and can be seen as a positive feedback loop. So when we look at the two loops, what we're looking at is we're looking at the combination of two signals that are going to ensure that we are feasting when we should be feasting and fasting when we should be fasting. All based off of the interplay between cortical structures in between the hypothalamus, higher cortical regions including the prefrontal cortex and the mammillary body, the caudate nuclei, as well as hindbrain sensors that relate to cranial nerves, modified by the endocannabinoidal system. The endocannabinoidal system uses endocannabinoids, and the endocannabinoids are going to modulate the receptors of leptin and ghrelin, insulin, and adiponectin. And by modulating the responsiveness to these receptors, we can change both feeding behavior as well as gastrointestinal functioning and energy balance. Some of the individuals that you'll see throughout your clinical career will have issues where they cannot lose body mass regardless of how they go about attempting to diet or exercise. And part of this has to do with sensitivity changes within the endocannabinoidal system, modulating the way in which these two loops lay out. We're going to have peripheral tissues that are going to come into play, such as the stomach, the pancreas, intestines, adipose tissue, skeletal muscle and bone will come into play, nervous tissue will come into play. Establishing what's referred to as the long loop. So the long loop is based off of energetic balance, in which we're going to have distinct peripheral signals. AMPK is one of the metabolites that starts to increase as we start having increased energetic demand. Ghrelin will start to increase as we start having increased energetic demand. Insulin will start to drop. Glucagon will start to rise. PYY will start to change. Leptin and adiponectin will start to modulate based off of sensitivity changes, where we should start seeing a reduction of leptin and an increase of adiponectin. All of these will feed onto the hypothalamus to start to change higher cortical functioning by changing release of the hormone NPY, neuropeptide Y. Secondarily, ghrelin, leptin, and AMPK can directly impact energy balance functions by modulating the rate at which growth hormone and thyroid hormones are being produced and released, as well as the relative sensitivity to insulin and glucagon at peripheral tissues when combined with some of the myokines, in particular irisin, IL-6, and IL-10. All of these modulations are going to drive the person to need to consume fuel sources. As soon as we start to consume fuel sources, we start to initiate the short loop. The short loop starts to get initiated as we start to have changes of the anatomy of the alimentary canal. This is going to stimulate the vagus nerve through the enteric plexuses, which will head back and start to stimulate the hindbrain through reflexive loops off of the vagus nerve. From the stomach, the pancreas, and the intestines, we start to secrete distinct endocrines that will modulate anatomical functions within the alimentary canal, such as CCK, gastrin, GLP-1, PYY, oxyntomodulin, and insulin. These two will head back towards the hypothalamus so as to induce the sequestration of eating, all based off of how the endocannabinoids are going to allow for stimulation or inhibition of the hypothalamic nuclei relative to leptin, ghrelin, insulin, and adiponectin signals. So let's take a look at this reflexive loop based off of the hormones that are coming into play and the hypothalamic nuclei that are going to initiate eating, or more importantly, not eating. So we have three real nuclei that are going to be coming into play here. We have the ventral medial nuclei, we have the paraventricular nuclei, we have the lateral hypothalamic nuclei, with two subset nuclei coming into play, the arcuate nuclei and the mammillary body. So as we start getting the signals of I need fuel sources or I'm hungry, ghrelin starts to increase, leptin and insulin start to decrease depending upon levels of sensitivity. So people who have metabolic syndrome, we will not see much of a change in leptin or insulin. We will start seeing a reduction of the pancreatic peptide Y or PYY. These are going to head to the ventral medial and arcuate nuclei and the paraventricular nuclei to stimulate the release of neuropeptide Y. Neuropeptide Y is going to induce a release of dopamine from the pituitary as well as from the caudate nuclei to head to the mammillary body as well as to head to higher cortical areas to induce the necessity to start eating. This whole reflex here can lead to eating issues because of the fluctuation of dopamine and off the next image, serotonin, due to the initiation of feeding. One of the things that you can determine as to whether or not someone is actually hungry or not is to give them something to do. A lot of the times, hunger issues get mistaken for boredom issues because of the need to get what's usually referred to as a dopamine fix. This dopamine fix is going to start with a spike of dopamine with the necessity to have a reduction of dopamine and a spike of serotonin. This is going to change catecholamine signals to alter behaviors where you're going to start searching for food and stop expending energy for things not related to eating. If it's true boredom, as soon as you start doing something, you get the immediate fix and you stop searching for food. If it's real hunger, you will continue to search for food. Sometimes just getting a bolus of liquid can go ahead and initiate the serotonin fix following the dopamine spike and initiate the reduction of want for food. This is part of the way in which the feel full mechanisms come into play by reducing the dopamine spike by providing the next step, which is the serotonin spike, which comes about when you feel satiated. It's usually associated with an increase of PYY, an increase of leptin and insulin, and a reduction of ghrelin, which heads to the lateral hypothalamic nuclei, which is going to cause an increase of POMC. POMC is going to reduce dopamine secretions and spike serotonin, which flips the prefrontal cortical response as well as the mammillary body response to desires, which means it's going to trigger a stop response within the seeking of food. It's going to trigger the stop response in terms of eating itself, and it's going to switch a little bit in terms of energetic expenditure, in which it allows you to go about expending energies for other things besides the desire that you had to initiate the behavior. When you're eating, you also get a spike of melatonin coming from the POMC, secreted from the pineal gland. When the cerebral cortex gets a spike of serotonin with a spike of melatonin, there is a change of cortical activity, an inhibition of the RAS, the glutamate pathway, and the noradrenergic pathway that leads to a rest time. If this meal is substantial, this can lead to a prolonged rest period. This prolonged rest period is what some people will reference as being the quote-unquote food coma that is associated with overeating. It's not necessarily overeating. It's just the meal was large enough to trigger a spike in both melatonin as well as a spike in serotonin. Now, the endocannabinoid system is going to come into play here. And what the endocannabinoid system is going to do is it's going to block the lateral hypothalamic nuclei while accentuating the signals from the ventral medial and the paraventricular nuclei. So what it's going to do is it's going to trigger you to want to continue to eat. It's not really going to fully block the stop eating, but what it's going to do is it's going to override the signals coming from serotonin. It's going to cause a small little spike of dopamine, and this small little spike of dopamine with a blocking of the signaling of serotonin will lead you to want to eat even though you don't need to eat. This activation of the endocannabinoid system is how some pharmaceutical cannabinoids will go about their function, leading to what some people refer to as people experiencing the quote-unquote munchies with exposure to tetrahydrocannabinoid, THC. We can use that for people who happen to have experiencing of wasting diseases, such as what we see with people who are experiencing AIDS-related issues, people who are undergoing chemotherapy, or older adults who do not consume enough because of the pharmacological changes that take place within the hypothalamus due to medications. So let's take a look at how ghrelin and leptin are going to go about causing the orexic responses, the need for food, and the anorexic response, the need for no food. Now, this change of orexia and anorexia will also change downstream hormonal secretions that are going to modulate overall energy balance. Leptin and ghrelin are the hormones that regulate basal metabolic rate. The books will tell you that it's thyroid hormones because of thyroid hormones' effect on mitochondrial metabolism. But leptin and ghrelin themselves is what will actually be modulating total energy expenditure as well as fuel intakes relative to energy expenditure. Now notice we have two distinct areas here. We have the orexic areas. Those are the areas that are going to need food because we have a negative energy balance. And the anorexic centers, those are the areas where we don't need food because we have a positive energy balance. Orexia is the process of eating. Anorexia is the process of avoiding food. Just because someone is having an anorexic response does not mean that they are anorexic. And that's something we're going to talk about in the next presentation. So we start with signals where we are fasting. When we start fasting, we start to increase lipolytic activity, in which the increase of lipolytic activity will have two distinct phases. It's going to change both leptin and ghrelin to reduce leptin levels and increase ghrelin levels. As we start doing this, we're going to have a signal that's going to activate the hypothalamus through our nuclei, our paraventricular, lateral, and arcuate, to stimulate the orexic centers and inhibit the anorexic centers. At the same time, because of the reduction of leptin and the increase of ghrelin, we get a secondary signal. The secondary signal is going to lead to a reduction of growth hormone and a reduction of thyroid releasing hormone and thyroid stimulating hormone. By reducing the thyroid releasing hormone and thyroid stimulating hormone, we reduce T3 and T4. By reducing growth hormone release, we initiate a change. Now, even though we say we're reducing growth hormone release, what we're actually doing is we're reducing the large growth hormones, the 22 kilodalton and the large molecular weight growth hormone, but allowing for these small molecular weight growth hormones to be released. What this does is this accentuates more lipolytic activity, but what it also does is it reduces the quote-unquote basal metabolism for the person. This is where excessive fasting will actually be counterintuitive to the weight reduction that is necessary for individuals because of the loss of expenditure due to the reduction in basal metabolic rates. Now we start to feed. As we start to feed, we start to change our lipolytic activities. We start to reduce lipolysis, which means we're going to start increasing leptin. We're going to start changing ghrelin releases. The change of ghrelin release and the change of leptin release is going to stimulate the anorexic centers and inhibit the orexic centers. This is going to allow me to have a fasting period. At the same time, we're going to start to see an increase of thyroid releasing hormone and thyroid stimulating hormone, which is going to cause the release of T3 and T4. The change of ghrelin, along with the positive energy balance that we get from changes of leptin, lead to the release of the larger growth hormones, which leads to an increase of tissue growth with the reduction of lipolysis. This leads to an increase of fat-free mass. When we combine the increase of T3 and T4 with the change of mass towards the fat-free mass, we start increasing basal metabolic rate. This is where allowing for multiple meals spaced evenly throughout the day can actually increase total caloric expenditure for people who are undergoing a hypocaloric or a calorie-restricted diet. Now, part of the problem here is that we have the endocannabinoid system coming into play. And this endocannabinoid system coming into play can cause secondary regulatory signals and can cause a prolonged sense of feeding, even though we don't need to feed anymore, due to changes of sensitivity to the leptin and the ghrelin signals at the nuclei within the hypothalamus. If that happens to be the case, what ends up happening is that even though we're attempting to stimulate the anorexic centers, we are still stimulating the orexic centers, causing additional consumption of food materials.